Okay, we're on. Gotta hang up. Bye. Well, I guess the first thing, um, kind of get an idea of who's listening, who's on. If y'all could just do a comment and say, for example, just put your first name, Cal, or something to let me know who is on. Now you can watch this at a later time. I guess one of the big things that I wanted to do was to um, make sure if you had any questions about any of the previous assignments or any assignment coming up, that you had a chance to ask those. And plus, we are going to be covering a few things that relate to some of the topics that you've already been working on, some of the review sheets and some of the worksheets that you've been working on. And as time goes on, we're going to be having other lectures like this where we're going to be covering new materials, like, say, for example, in ecology and some things having to do with plants. And so that is going to lead up to the um, possibility, and actually pretty good possibility, that we're going to be having some tests coming up. Um, there's several ways we can do this. There is one way that we can do it where we use some programs that Cleveland State has that would allow me to give you a test that is pretty much individualized to you. In other words, I would get a bank of, say, 100 questions, and they would give you a 50 question test. From that, I'm not sure we have to do that. I think the simplest thing would be to simply trust you that when I give you a test, that you're going to work through it on your own. And in these early tests that we might have coming up, and again, these are all just possibilities. The early tests coming up, we may actually let you use the worksheets and review sheets that you have been working on, and that way you can use those for that. Now, I'm not getting any comments from anyone. And like I say, this is all new to me, and so uh, I'm assuming that when you make comments, they're going to appear on here. If not, then I'm just going to go ahead and keep lecturing, and we'll see what happens there. Like I say, I'm learning this as as you are too, so that's just part of it, I guess. Now, um, I guess to go ahead and just get started on this, you've already done a um, review sheet and so a lab handout where you did some drawings on mitosis and meiosis. And I want to tell you that y'all are getting things into me really, really well. I've gotten most papers back from almost everyone. There are a few folks that have turned theirs in or tried to turn them in by Google Docs, and it may be my fault. I've tried a couple of times to get on there to receive those, and they have denied me each time. And I may check with Mr. Patton in the next day or so and see if I can find out what the problem is. And like I say, it's probably at my end, and I'll try to fix that. But if, especially for the first things that you're doing here, if you will just simply photograph them with your phone, and I'm asking you to photograph them straight down. When they're at a slant, it's hard to read them. If you go straight down and get a good big picture with your phone, maybe turn your phone sideways so you can get a good view of the whole page. I can even make copies of those if I want to. I've already tried that, and that does work. So that's fine for these assignments that you've been doing: mitosis, meiosis, the review, and DNA and RNA. Um, those are all very, very adequate there for that. Um, when you do the virus report, now I've had some questions about page numbers, and I think I found the problem. I used when I put those page numbers down, I used a previous edition. I think the edition I used was the eighth edition. I think you've got the ninth edition. So I found that problem, and I can fix that. That's not a problem. But I think you were able to figure out that you started reading at 205, and I think you figured out where that figure was for the virus. The coronavirus is a part of a family of viruses. The COVID-19 is one member of that family. And one of the things that makes them unique and also makes them fairly easy to get rid of is that their outer coating has a glycoprotein. Those spikes that you see on pictures of that virus, that's a glycoprotein spikes that are on those. And when that is exposed to things, and that, by the way, is a type of lipid. And so lipids are fairly soluble in things like alcohol. So alcohol swabs, isopropyl alcohol can get rid of those. The one of the big ingredients, one of the big active ingredients in these little pads that you get from in grocery stores when you go in to swab your cart down, the handle of your cart, that has alcohol in it. The Germex, various Germex mixtures that you see have alcohol in them. And so that helps get rid of the virus. In addition, heat will cause the glycoprotein spikes, those spikes, to degrade, and that will cause the virus to die as well. Now, this also leads us to talk about the idea of vaccines. There are a bunch of people who are working on vaccines. Probably better than 20 groups are working on various kinds of vaccines. Some are in more advanced stages of testing. There is a group, I think, out of Johnson and Johnson that is working on a kind of vaccine where they are in what they call stage 2 testing, which during stage 2 testing, there are some things that they do with humans. It's not extensive testing with humans. Don't get me wrong there, but there are some indications that they may be able to have a vaccine by the end of this year, and that's amazing. Usually, vaccines like this take at least 2 years and a ton of money. Now, in some cases, over $2 billion to get a vaccine that's viable, that's safe. Now, no vaccine is 100% safe, but viable and able to help people. Whether they can speed that up, I don't know. FDA, the Food and Drug Administration, has been working to shorten the process by which vaccines can be made and approved. Whether that is going to take place, I don't know. But I would suspect that they're going to be getting that real quick. Now, I'm not getting any comments. I don't know if there's something that I'm not doing correctly. My phone is right here next to me. I know Cal knows my number. If there's something that I need to be doing to add ability to put comments on here, y'all let me know. My number is 261-2635. And if anybody can think of something I need to do there, or if anybody wants to ask a question, I'll get about 15 calls now. But the virus report is to try to get you familiar with this. I think in a biology class with something this life-changing that is happening to our country, that you should know something about this. And I think that's going to help out. Now, we see some indications that the virus is beginning to not go away, but it's you keep hearing them talk about flattening the curve. Well, flattening the curve is where they're talking about the increase in the number of people that are dying each day. In places like New York, which some folks think is about 2 weeks ahead of many other places in the country, the uh, the number of people that are being admitted to hospitals and number of people that they think are actually exposed to the COVID-19 is beginning to the rate of increase is beginning to decrease. In other words, if you're plotting this on a graph, it is approaching the top of the curve, and then in some respects, it may even be going down in the next few days. The number of dead is also the number of increase in deaths is also decreasing. Uh, there is another aspect of this that is very interesting, and that is that the number of people that have been exposed to this virus may be vastly more than what we originally thought. In fact, there may be, it's what is called a herd immunity. And the herd, which is us, um, builds up immunity in two ways, really two primary ways now. And one is by having the disease and actually showing symptoms of the disease. The other way is by having the virus infect us, but we show no symptoms. And that is a very definite possibility. And the number of people that are actually fitting that second category, we think is going to go up dramatically as time goes on. We don't know that yet, but they're developing some tests, and you all know what antibodies are. And so when you have a virus like this, your body is going to develop antibodies against it. And if you have a way of detecting those antibodies, then you can detect that the person has been exposed to the COVID-19, even though they may have never showed symptoms of it. And so when we look at the number of people that have been exposed but have shown no symptoms, and add those to what we already know about that have shown symptoms, the number that have not shown symptoms may be double or triple what we see as those who have symptoms. And if that's the case, then that means that we are developing a herd mentality or herd immunity. Now, the hope is that would be taking place. Plus, if a vaccine comes along, that they'll be able to give people a vaccine, much like they did in smallpox, that the vaccine is giving almost universally, and that then adds to the herd immunity until eventually, and by the 1960s, 1970s, certainly by the 1980s, people were declaring that smallpox had been eradicated. Now, whether that takes place in a short period of time is probably not possible, but it's something that would be over the next year or two. This virus is probably not going to go away. Now, saying that, it probably, we have no idea if it's going to go away for sure. But one thing that we do know is that it has the ability to mutate. Now, the virus that caused the flu epidemic of 1918, we think maybe started in 1917. Where it started is open to debate. But the point of the matter is that by the end of 1918, and certainly by the beginning of 1919, in the early months of 1919, it basically disappeared. Now, how it did it or why it did that, we don't know. But a good speculation would be that it mutated to the point where it no longer affected human beings. It was actually one that we think came from pigs. Now, the COVID-19, the thinking right now is that it probably came from bats. Now, to understand that, you've got to understand that in China, it is very common to have meat markets, and I've told y'all about this in class before, that meat markets where the meat is basically, the animal is basically hung on a hook, and the shopper walks by where the animal is hung on a hook and says, "Oh, I like that animal," and they take it off the hook, and they get blood all over their hands and everything, and that then they take it home and they expose their family and they expose everybody else, and I think that's how it may have got into humans. And then in humans, it developed or already had this possibility. It already had this, but it developed the ability by mutation to actually transfer by uh, through the air. And so that people going through Walmart or stores or out in the public nowadays, they're wearing masks. The masks are not 100% effective, but they're better than nothing. And one of the main things is that the mask prevents some of the transmission or some of the water droplets that you breathe out that are going to infect other people. So that's again, it's not 100%, but it is something that does help. It's anything like that. Even a covering a bandana over your face is going to help some in some respects. Even the medical masks are not 100%. They're in the 90s. I've seen up as high as 95, 98% effective, but it's very hard to find something that's 100% effective against viruses. So the point is, and by the way, this is one of the reasons why that you recently heard, I assume all of you have heard, that school has been canceled in the state of Tennessee and for this school year. And I hate that, especially for those of you who are seniors, that this is, I can tell you this much, that Denise Watson has been very adamant about wanting to have a prom and wanting to have a graduation for you. When that might happen, I have no idea. Would it happen in June? It is very doubtful. July, it is more possible, but we just have to see how things are going to go in terms of that. How the virus, like I say, a lot of their projections about this virus is it was projected that 100,000 people would die or more in the United States. They projected downward now. Their computer models that they use are now predicting less than, certainly I would think less than 50,000, and in some respects maybe less than 30,000. That's still a lot of people. Don't get me wrong. I'm not belittling that at all. But the severity of this virus is we're finding out probably not going to be as severe as what we had once thought. Now, having said that, what's going to happen in the future, I don't know. This virus, there are two kinds of nucleic acid material that are in the middle of a virus. One is RNA and one is DNA. And this is where when you study DNA and RNA and how DNA, what DNA does, DNA can replicate, DNA can make copies of itself, and DNA can also make RNA. Well, in the case of a virus that has RNA in it, we oftentimes call it what's called a retrovirus. And this kind of virus is actually able to, the RNA is able to go into the cell and reverse the process. Instead of DNA making RNA, this RNA actually has a process by which the end result is DNA. Then the DNA uses the mechanisms of the cell, the things that are in the cell still living, that it uses them to then make RNA and make the new viruses. Okay. Now, having said that, DNA inside of a cell like that, and RNA has the potential to be changed. And this is what we'd call a mutation. And so if the mutation changes the RNA, the DNA, whichever it is in the virus, enough that it could cause the virus no longer to be pathogenic to people. It no longer hurts people. It's no longer virulent. And that may be what happened in 1919 with the flu epidemic of 1918. Now, the flu epidemic of 1918, according to many estimates, killed at least 20 million people. Some estimates might put it as high as 50 million people. We really have no way of knowing. So many people died and their deaths were not even reported. My father lived in Maryville at the time, and 1918, and that was the epidemic was hitting at that time. And he said it was so bad that people would take bodies out to the side of the road, and they'd pick them up in wagons that came through. Now he remembers seeing that. Now we think that my grandfather actually had that virus, and they put him in a shed behind the house, and they fed him in a little slot, a little opening under the door, and he got over it. And that in the virus of 1918, it had a fatality rate, a rate of actually killing people that far uh, is by far is more than what this virus is now. At least half, if not two-thirds of the people that contracted the disease died. And like I say, we don't know the exact figures because records weren't kept for a lot of things like that. We think the virus COVID-19 has a death rate or fatality rate of 1% or certainly less than 1%, especially if you consider all the people that have had the virus and not shown symptoms. Now, again, I'm not trying to belittle the COVID-19 or belittle any of the things that we've done to try to keep people safe. That should be a primary concern of ours. But like I say, in 1919, this virus basically went away. We didn't even know what it looked like until they found some slides uh, that were stored away of soldiers' lungs that had had this disease. And they've got a picture of it, and they've actually got samples in the Atlanta CDC. At least I assume they have it at Atlanta CDC, way far underground in extreme security. But they didn't even know that until, say, about the 1990s or the early 2000s. So that's been a mystery type disease. We have learned a tremendous amount about the COVID-19 virus, and like I say, it is sensitive to heat. It is sensitive to soap. Soap will dissolve the coating on the outside of the virus, and that'll kill the virus. That's the reason they want you to wash your hands, suds up the hands real good for at least 20 seconds. And alcohol swabs and Germ-X. I can't give a 100% kill rate, but they're certainly going to be better than nothing. All right. So I'm not, again, I must be doing something wrong. I am not getting comments. I don't know if I did this. Okay, I just got where it looks like there's 12 people watching or 12 people signed up or something. And maybe nobody's doing a comment. I don't know. Let's see if I can click on something here. Nah, that doesn't help me any. Show custom. Okay, like I say, I'm learning. I'm not an expert on this at all. I thought maybe you could comment. I thought that was possibility, but maybe it's not. Oh, there's a comment. Caden says, I'll comment so we know it works. Yeah, it's working. Okay. So if you need to ask any questions, now is the prime time to do that. Now, my next lecture, I'm not finished with this one yet, but my next lecture will probably be tomorrow. And again at 9 o'clock. And I eventually want to make this a regular thing. Yeah, McKinley, it is working. I do see your comment. A regular thing every Tuesday and Thursday. Now, if that's not going to work out, let me know. But that's what I'm thinking right now. Hopefully, I have given you enough information that you can know a little bit more about how to do the virus part of your assignments coming up. And if I don't have the instructions in front of me, but I think that particular assignment is going to be worth a test grade, if not 2 test grades. If my memory serves me, there is a part of it that is basically written out that you answer questions, and there are some drawings that you make, and there's also a part of it where you do some grafting. Now, again, the graph paper is something you can download off the internet. Certainly don't have to go out and buy it. You can download tons of it. That you've used at school for me, I downloaded off the internet and just printed it. That works fine. Just remember your graphs have to have titles, and remember that you have to label what the y-axis is and the units, and what the x-axis is and the units through it as well. And I think in the instructions I gave you what those things are, and so hopefully that's enough for you to write that out. And when you do that, just make a photocopy or a photograph of it with your phone or iPad or something, and have it pointed straight down. And I can make a copy of that then, and I'll read it over and see how you did. And if everything's correct, then you'll get a 100 good grade. If it's a few things I think needed to be different, then your grade will go down a little bit. Now, let me tell you something about grades. I don't think it's a secret. Your college grade is going to be separate from your high school grade. You know that. We've been through that in the last semester. And your high school grade, if you go on TeacherView, I have posted my grades probably a week ago or 2 weeks ago. All the teachers are supposed to post their grades by this Friday. I've done that a good 2 weeks ago, I think. And so what you see there is your third 6 weeks or third 9 weeks grade. And you can't, for a high school grade, you can't make below that grade. I hope you understand that. But you can certainly improve your grade. If you're doing all these assignments and do all the things for biology 2 that I ask you to do, your grade is going to improve. I'm going to tell you that right up front. Now, the college grade, I talked to a lady who's in charge of, I think she's actually in charge of all the dual enrollment biology. Very nice lady. And she's the one that actually got me thinking about that. I had been thinking about doing this, but she said, yeah, several of the teachers are doing things where they lecture to their students. And I said, oh, okay, if I can work on YouTube, then I can do that. Okay, now this is working out, I think. But the point is that your college grade is going to depend upon these assignments you're doing. And each time you do an assignment like this, I'm putting down a 100 quiz grade. And if you do good on the virus, then you're going to get a good grade on that. Now, coming up on test, again, if you have a test in front, it's just like any test on anything else, that if you have a test grades are going to, you could make 100 on them. I don't know. I mean, I think it's entirely up to you. Depends on how we do this. But that is, again, going to count toward your grade. Okay. All right. Now, I hope that has helped in answering some questions. I'm going to go a little bit into DNA and RNA at this point. And if you wanted to take notes, that's fine. A lot of the things that I'm going to be going over are on that DNA and RNA review. I'm just going to be going over some things that just kind of emphasize some things that are, in most cases, already on that. And so I think that's a good place for you to start. This maybe just reinforces some of those things. Back in the 1950s, you have to understand something, that scientists were very, very competitive with each other. That is, you think of that in sports, but you don't think of it in terms of scientists. The scientists are extremely competitive, and they want to be the first to do something. And so people wanted, by the 1950s, it was pretty clear that chromosomes were very, very important in how cells were controlled, and that chromosomes were made of DNA, and therefore that if we knew what DNA was or how it was made, then we could have some idea of what to do with that. And a bunch of folks got involved in this. Now, Watson and Crick are by far received the most credit. Rosalind Franklin probably should have received more credit than she got, but nonetheless, she was very instrumental. Did a lot of X-ray uh, photography type work that then was passed on to Watson and Crick. And you think, well, now why do they pass things on if they know it's going to help the other group? Well, that's the way scientists are. That's almost like a code of science, that what you've learned you try to pass on to other people, other scientists. And so they use what she developed and eventually came up with a structure of DNA which they called a double helix. Um, it's we now know a lot about the chromosomes that make up the 46 chromosomes in each of your normal body cells. We know where a lot of genes are, and we know in many cases what those genes do and how the chemistry of them works. You see, we understand that DNA is in the nucleus of a cell. Now, I understand that there's DNA in chloroplast. I understand there's DNA in mitochondria. And at this point, we're not talking about that kind of DNA. We're talking about the DNA that is in a nucleus. And this is a eukaryotic cell. And this DNA can make copies of itself. We call that replication. It can also make RNA. We call that transcription. Okay. And that RNA is there's three types of RNA. There is messenger RNA, transfer RNA, and ribosomal RNA. The ribosomal RNA we're going to basically kind of ignore. Obviously, it's found in ribosomes. Messenger RNA is just what its name says. It is a message that goes out of the nucleus into the cytoplasm of the cell and specifically to the ribosome. And with the transfer RNA bringing amino acids to the ribosome, excuse me. You know, when I talk a lot and I lecture, I sneeze. It doesn't mean I have the COVID virus or anything, so don't get all panicky. But the besides, I'm here by myself. But anyway, the messenger RNA is the message for a protein, and the transfer RNA brings the amino acids. What, Kendra? Gasp. Why are you gasping? But the transfer RNA brings the amino acids, and then at the ribosome, these are connected together to form proteins. And it's these proteins. Witt's going to die. No, I'm going to die. I will die eventually, but not tonight. I don't think so. But anyway, these proteins then control what the cell does, and these proteins can be extremely complex. You remember me telling the story about a symposium I went to several years ago, where a professor or a researcher, protein researcher, a biochemist was speaking, and he said he had put his data into the supercomputers at Oak Ridge. It took about a minute and a half to give him the results of his data, and he told us, he said it's going to take him the next 3 years to figure out what that data says. I'm going to ignore McKinley. She's a morbid soul. So that means that you have in your cells extremely complex proteins, and if there is the slightest problem with them, they don't work. We saw an evidence of this when we were studying Tay-Sachs disease, and it was a mutation that affected four atoms on the DNA, and that then caused the person to have severe physical problems, and in most cases died at a very early age. But that just illustrates that and this also goes back to evolution. One of the biggest problems that evolution has is the idea of information. DNA is information. And in order to have a new species, you got to have new information. You can't just scramble up or reshuffle the information that you've already got. You've got to have new information because you got to have new proteins. And that's one of the biggest things. We don't know how that happens. Mutations are just not common enough to account for all the changes that evolution says should have happened. And even if you look at mutations, we got to understand that most mutations are going to be harmful. And a high percentage of them, one might even say 80 to 90% of them are going to be fatal. I mean, all you have to do is look at the research that's been done with fruit flies. Ever since the 1940s, they've been exposing fruit fly populations to X-rays and causing tons of mutations. They've never made a better fruit fly. In all the cases that I know of, the mutations that were made actually were harmful. You say, well, wait a minute, Mr. Witt. They made a fruit fly that had more wings than it was supposed to. Well, that actually made it fly less than it was supposed to, and so that didn't turn out to be an advantage. So this is one of the big problems that we run into when you look at in terms of mutations, how these mutations are going to affect organisms. Okay, I tell you what, y'all have stuck with me real well. We're going to stop there. I'm going to end this. We're going to pick it up tomorrow again at 9 o'clock. And what I want you to do is to have your book with you. I want you to have your notebook with you. You need to have the evolution sheets with you, the DNA sheets with you. Mitosis and meiosis, I'm not going to go over very much at all, so you don't necessarily have those. And you need to have any questions that you have. Oh, you don't have your book. That's a bad thing, Caden. Maybe you can borrow one from Michaela or something. Some other people don't too. Oh, well, okay. Well, most of this stuff that you have on these reviews, you can look up on YouTube. I mean, not YouTube, but Google. And like I say, you need to have also the ecology handout that I have just posted this past few days. And I'll go over a lot of things in there. And so probably the first test that we're going to have will be coming up next week, and we'll go into a little more detail about what that's going to be. And I'll probably let you use your review sheets. That doesn't mean that the test is just going to be question and a one-word answer. You'll probably have to be a little more, stretch out your answers a little more. And plus, you're going to have some true false, which y'all hate true false, I know. But that, I think, makes you think. I guess that's the reason you hate them. But anyway, I hope that all of you are well, and you don't have to just be on the live stream. You can tell people that have not seen this that they can look at it. It's right now coming up on 39 minutes, so it's not a hugely long thing. Each time I do this, I'm going to put it under Witt's lectures. Witt's lectures. As long as I don't think I have to change that. Yeah, you about hating. I know you do. So anyway, I hope y'all have a good evening. I'm glad you joined. I appreciate that. And y'all just keep turning these things in to me. Again, making photographs with your phones, and then just email it to me. I am getting those. I am recording those. The virus test graphs will be a test grade. Yes. That there's going to be 2 test grades coming out of that. Now, like I say, folks, you can really improve your grade tremendously by doing all these things. I'm not trying to kill you with all these assignments, but you do have definitely an opportunity to really help yourself. High school grade immensely, and your college grade, I think, can only go up. So, and again, college is going to end here pretty soon. I'm not sure the date that I don't know if they've extended that, but I suspect that I'll have to have grades in by somewhere around May 15th, if not maybe close to May 20th. So we don't have very many days ahead of us. We've got about a month, and so we're going to try to take as much advantage of that as we can. So again, have those things in front of you tomorrow night at 9 o'clock. And I appreciate you joining us here. Is it okay if the statistics we find are not completely accurate by the time you go? Certainly, certainly. Um, that's so. What are you thinking about a final? I honestly don't know. What we have when I talked to the lady from Cleveland State, they are going to try to have a final. We are required in college to have a final. How that will be, what form that will take, I don't know. Um, like I say, there are some options. I think right now I'm leaning. Now I'll be honest with you, I can change my mind on this, but I'm leaning to do it the same way we've been doing these assignments, so that I post it to you as an email, and then you record your results and you photograph that and turn it in to me, and then I can grade that. Now that's what I'm thinking right now. Um, Whether that is going to be the way it actually happens, I'll be honest with you. I'm still learning about this stuff, and if I can find a better way to do it, I certainly will do that. But that's what I'm thinking of right now. Are there any other questions that anybody has before we sign off? Well, again, it looked like the comments worked, and hopefully uh, I guess this number that's in the upper left-hand corner is the number of people that are watching it. I guess you can subscribe to this. I don't know. Those are all things. This is all new to me. So y'all have a good evening. I'm going to sign off now. Thank you for watching.